Our final speaker in session 3, talking about nanomedical processes and development today, is Dr. Mark Kester, who we've heard from a couple of times already with some very astute questions. Dr. Kester is the G. Thomas Passanati Professor of Pharmacology at Penn State University College of Medicine, Hershey, Pennsylvania, and Director of the Penn State Center for Nanomedicine and Materials. Dr. Kester is also the co-author of Integrated Pharmacology, published by Elsevier, which has been recently recognized as one of the most highly commended textbooks, one of the highly commended textbooks in the field by the British Medical Society. This afternoon, Dr. Kester is going to speak on dihydrosphingosine 1-phosphate and how it triggers linkage, dihydrosphingosine 1-phosphate triggered linkage, switching of myeloid-derived suppressor cells and anti-tumor immune effectors during photoimmune therapy. I'm really looking forward to your talk. I remember listening to you previously, learned a tremendous amount. Dr. Kester, please join me at the podium.
First, I'd like to thank Martine and Barry. Haven't seen you in a year, and by the way, I'm glad to see you, because without Genesis' help and Temple's help today, driving through the snowstorm and checking on me every half an hour to make sure that I didn't take the four-wheel drive and drive it off the side of the road. So usually I say I'm happy to be here. In this case, I really am happy to be here. So let's go to the first slide, because Martine talked about dihydrosphingosine 1-phosphate. Obviously, I've been giving lipid talks in the last couple of weeks. And for this talk, we're not even going to talk about the lipids.
Oh, okay.
Isn't that much easier?
Well,
Because I want to talk about a therapy.
Photoimmune nanotherapy is a great idea.
I want to talk about a therapy. I want to talk about a therapy that uses nanotechnology, that upregulates the immune system and makes a theranostic, which is the word that the NIH is using right now. Is a therapy, it's an imaging agent, and a diagnostic all in one.
Great.
And I'm going to show you how to commercialize it and how to try to get it from the bench to the bedside. I'm a faculty entrepreneur, so basically I do work for Penn State College of Medicine. However, let's see, next one. That's not the right way. I also have co-founded a company, and this is my slide to tell everybody in conflict of interest that I'm Chief Medical Officer of Keystone Nano, and I formed this company with Jim Adair, who's the National Science Foundation head of the Particulate Materials Center, and he was a nano geek. And he came to me and said, I can make deuterium, I can make gold, I can make cadmium, selenite, and zinc. And I go, and what do you want to do with it? He goes, I want to cure cancer with it. I said, I'm not an IBM computer. I'm not cadmium, I'm not selenite, I'm not zinc. Can't you guys do something simple? Can't you do something out of a biodegradable, biobenign compound? What about calcium phosphate? So I'm going to show you how calcium phosphate, and there it is right there, can be made on the nanoscale and be used to encapsulate and protect. And what I'm not going to talk about is small molecules, say like doxorubicin, or small interfering RNAs for molecular-based therapeutics, but I'm going to try to show that we can actually harness the power of light for theranostics, truly to use light to kill cancer cells. So with that, just for a background, we now call these materials nanojackets. Why? Because Nature wrote a nice article about us, and they used the word, and I said, all right, that's great, because they're dressed to kill. And that is one 20 nanometer size nanoparticle of calcium phosphate. And what makes this particle different from all other nanoparticles is it's amorphous. It's, for lack of a better word, porous. We can put the active ingredient inside the nanotechnology. A lot of nanotechnologies are solid, and you put the active on the outside, so it can be degraded, metabolized. It's not going to work. We actually put the active inside the nanomatrix. And here we have an example of 5 different fluor probes inside the nanomatrix, inside the calcium phosphate nanoparticles. And we've published now in ACS Nano Letters over the last year and a half that these materials are biobenign, biodegradable, and we'll talk a little about that right now. So what also makes these particles unique is they're biosmart bombs. We've engineered them to be stable at pH 7.4. In fact, those fluoroprobes you saw before are now going on 2.5 years. And if you've ever taken a fluoroprobe, put it on the desk, within a couple of days, let alone a couple of hours, they photobleach, they photodegrade, they no longer fluoresce. Our materials can stay inside these nanoparticles for 2 years and counting and still fluoresce. So we truly get that protection. But what's nice is they're stable at 7.4, but they're designed to dissociate, to dissolve at pHs below 5.5. So we take advantage of the cell's ability to pinocytose these materials, which are 20 nanometers in size, non-aggregating. They actually end up into the cell through pinocytosis, and as they end in the early endosome, as the pH falls, and we've now published this in 3 different publications, the calcium phosphate matrix dissolves, out comes your active. The phosphate now acts like a buffer. The endosome doesn't like to be buffered. It explodes. Out comes your material into the cytosol. And if it's a small molecule or siRNA, that's exactly where you want it. So we now have a trigger that we have designed inherently in the calcium phosphate nanoparticle. Here's a TEM of the calcium phosphate nanoparticles. Notice they're non-aggregating. They're not agglomerating. We can actually make a solution of 10 to the 15th particles per cc. That's 1,000,000 billion particles. Surprisingly, that's not a lot of calcium. When we actually deliver a bolus systemically through a tail vein, in, let's say in a rodent model, we're actually giving less than 10 to the negative 7th molar calcium, which is 3 orders of magnitude below systemic load. A lot of particles, not a lot of calcium, but a lot of drug and or active ingredient. Notice these particles are 20 nanometers, plus or minus, very tight distribution, and they don't aggregate because we can actually passivate the outside of these surfaces with pegylations, etc., so they act like Teflon and they don't aggregate among themselves. They don't become macro and become lunch for a macrophage. So those particles we just saw before were actually loaded with indocyanin green. It's a near-infrared photoprobe. It's FDA approved, and it's used for imaging of the peripheral posterior segments of the eye, and it's also used for other medical indications. What we've done is inside each of those particles, we've put indocyanin green. What are the limitations of indocyanin green, and what do I want it to do? Well, the problem is it's relatively unstable, it dimerizes, it quenches, it basically binds to proteins, and the plasma half-life is on the order of minutes. When we have our materials in systemic circulation, we get half-lives of 18 hours. Huge difference. The benefits of the encapsulation is the caging within the calcium phosphate nanoparticle prevents agglomeration. Basically, we get photostability by putting pegylations on the outside of the calcium phosphate nanoparticle. And we can actually move these indocyanin green molecules into the tumor, as you're going to see momentarily, to allow imaging of tumors. And you'll see we can go as far as 6 centimeters. You start thinking mammography at this point. So I'll just give you a little of the biophysics beforehand. We had to make these materials stable in physiological solutions. A lot of nanoparticles are great, but as soon as you put them in physiological buffer saline, let alone the body, they self-aggregate, they agglomerate. Our materials are delivered in PBS or equivalent and do not agglomerate. Here's an example of up to 288 hours of these materials in PBS, and you can see the size doesn't change. And this has all been published in ACS Nano, so we'll go quickly through this first 5 or 6 slides. Notice we get an increase of the fluorescence when we encapsulate the indocyanin green. And we actually use fluorescent correlation spectroscopy with some of our partners, Peter Butler, Peter Eklund, and we get quantum efficiencies of 2 to 5-fold when we encapsulate because we don't get the photo degradation and or the photo aggregation. When we actually measure, we take a laser and we actually excite these calcium phosphate nanoparticles, and we're going to use this in the photodynamic therapy coming along. We see that when we encapsulate these indocyanin green photoprobes within the calcium phosphate matrix, that's the blue, compared to free indocyanin green, which is just indocyanin green without being encapsulated, you can see at both wavelengths, power intensities, you get increased fluorescent lifetime of the indocyanin green. You protect it within the calcium phosphate nanoparticle, and for any amount of energy going in, you get multiple bangs for the buck of energy coming out. The other thing we've noticed, that if you took indocyanin green and you actually measure the fluorescence in different medium, let it be H2O, DMEM, PBS, DMSO, the wavelength shift is very, very unstable. This is the free indocyanin green. When we put the indocyanin green in the calcium phosphate nanoparticles, the curves superimpose, again showing protection of the indocyanin green within the calcium phosphate matrix. So what can you do with this? Everybody talks about enhanced permeability and retention, the ability of a tumor to grow quickly, want to form a vasculature. They form a vasculature very quickly, but not that well, and materials come in, but they don't come out. This is an example of using photo imaging with the indocyanin green to actually look at enhanced permeability retention. All our animals, we got 4 animals, they follow them over time, 15 minutes, 3 hours, 24 hours. He moved over here. We have another picture where he didn't move. 48 hours out to 96 hours. Animal 1 just got ghost calcium phosphate nanoparticles, just a calcium phosphate nanoparticle, no fluoroprobe. Animal 2 gets the free indocyanin green, just the indocyanin green, not encapsulated. Animals 3 and 4 get the indocyanin green within the calcium phosphate matrix. The 4th animal, that matrix, that calcium phosphate nanoparticle, is highly pegylated, which will increase the T1 half of the material. Let's go down to the 96 hour. These 2 little headlights over here, that's the tumor. All the other material has been cleared, tumors are lighting up. And these are systemic tail vein injection. We're not injecting into the tumor. This is one injection into the tail vein, and over time it accumulates. Because you can see as you go back, you can see the tumors as early as 24 hours. The other thing you see is look very quickly. You start seeing the material, that's the liver. The material goes to the liver, but doesn't accumulate in the liver. And because of size, it actually ends up going through the hepatic biliary recirculatory tree, and you can see over time we've actually pulled out the intestine and we've actually published this, but the material ends up in the poop, in the feces. So this is a nice way to get rid of the material. Nice clearance mechanism, and it does lead to minimal liver toxicology, and only 5% of the material goes to the renal system anyway. Okay, so now we can actually see the tumor by enhanced permeability retention. Here's one more example, another animal, again a nice example of the only thing lighting up is the tumor. We've then taken the calcium phosphate nanoparticles and we've put in gold. And now we use EM to look at the gold calcium phosphate nanoparticles. And you can see, you might have turned down the lights, but you can see the gold encapsulated calcium phosphate nanoparticles can be picked up. They're not agglomerating, they're in the endosome, and that's where they're going to dissolve. And that's an example, one of the proofs we have that these materials can deliver cargo by this pH reduction. This is what we call the Costco experiment. We actually went to Costco, one of the large purveyors in the United States of food, and we bought a pork butt. And basically, we actually put in the indocyanin green particles, either free indocyanin green and or indocyanin green within the calcium phosphate nanomatrix. And we can see in this example, you can see up to 3 centimeters through biological tissue. In the experiments we're now publishing right now, we're going 6 centimeters. So that's talking mammography, we're talking the ability to image through biological tissues. And the reason we can do it is because we get this quantum efficiency, this increased bang for the buck, because we've encapsulated within the calcium phosphate matrix. That's all passive targeting. This is actually going to be now published in a week and a half in ACS Nano, again with an editorial. This is now, can we actively target the calcium phosphate nanoparticles to the tumor? So what we've done here is we've done it 2 ways. The left side of panel B is a strep avidin biotin mechanism to put an active agent on the outside. The right animal here is we're using a maleimide chemistry. And what we're doing here is we're either putting pentagastrin or decagastrin and coating the outside with the 5 or 10 amino acids of the gastrin molecule. Why? Because some of our collaborators, such as Jill Smith, who's an oncologist, has shown that pancreatic cancer, but not normal pancreas tissue, expresses and synthesizes and releases gastrin and has upregulation of gastrin receptors, particular to a specific small part of the gastrin molecule. So what we're seeing right here, and you can see really here at 24 hours, that spot That's the pancreas. When the material was not actively coated with the active targeting moiety, the pancreas doesn't light up. When you put the gastrin on the outside through bioconjugation, you now light up the pancreas. There's the pancreas, that's the pancreas. These are the stomach too. That also expresses some of the gastrin receptors. So we can actually now move the nanoparticle to the tissue we want to go to. So let me spend the last 5-6 minutes on what we can do with this. This is all about imaging. But I said we want to be a theranostic. We want to do therapy and we want to do imaging at the same time. So let's go into what's called photodynamic therapy. And this is one of those serendipity type of experiments that basically go, I'm glad sometimes people make mistakes. So Brian Barth, who's one of my senior postdocs, basically came to me one day and said, Dr. Kester, Mark, you've got good news and bad news. I said, well, okay, what's the bad news? Well, we usually do experiments, as you saw, for 96 hours. And then we just want to do the imaging, and then we sacrifice the animals. And we have multiple animals experiments going on in real time. He said, well, we forgot one series of animals, and basically they went on for a month and a half. And you got to pay the bill. I said, what's the good news? They don't have tumors. So that we started thinking, can we use this enhanced quantum efficiency of the light to actually kill tumors? The technology is known as photodynamic therapy. It's used for esophageal cancer, bladder cancer. It uses a photosensitizer, light, and oxygen. The photosensitizer is excited, and basically you form a single molecular oxygen species, which then is activated in the single oxygen state and makes reactive oxygen species, and you kill the tumor from the inside. The problem with this therapy is basically the photosensitizers don't work. They have very low quantum efficiencies or quantum yields. The short half-lives are in the order of minutes. You have lack of specific targeting, inability to penetrate the targets, and basically the mechanisms are not well worked out. And what's not here, these tumors are hypoxic for the most part anyway, so you're not going to have much oxygen to make reactive oxygen species. What we're banking on is the fact that because we got the quantum efficiencies, because we got the protection, because we got the increased half-life, because we can target, maybe we can resurrect photodynamic therapy. And in that process, we came up with photoimmunonanotherapy because we see we're going to be working through a whole new mechanism of how these photosynthesizers work when they're encapsulated. So let me just go right to the chase. Does it work? So here we have two different mouse models. This is a nude mouse model. It's a breast cancer tumor. It's an MDA, highly aggressive estrogen-responsive tumor put into the hind flank of the animal. Here we have a syngeneic model. We're putting a mouse tumor inside a mouse with a complete immune system. We'll come back to that in important. I just want to go down to this green line and this green line over here. What we're doing is we're taking the calcium phosphate nanoparticle with the indocyanin green encapsulated. We stick a bolus into the tail vein for 24 hours. We see within 24 hours that the material ends up in the tumor. At that point, we take a very good photo laser and we actually shine it at the tumor. And we can actually focus the laser. If it's an orthotopic model, we can focus it inside the animal. If it's on a nude mouse back, we can focus it on the tumor. So it's one injection, one shot of the light. And then we follow the tumor over time. Here are all the controls: PBS, ghost particles, free indocyanin grain, calcium phosphate nanoparticles with indocyanin green but not have the high pegylation. All the tumors growing. This is the indocyanin green calcium phosphate nanoparticle, highly pegylated. Tumor doesn't grow. All in the nude mouse model, but also in the syngeneic model. Bottom line, we made a theranostic: image and treat. We actually went, that's a solid tumor. We even made it harder. We said, well, can we do it in a non-solid tumor model? This is a model of leukemia. It's a CML model. It's basically, we've extended the lifespan of this animal about 30%. And here we're actually focusing the laser on the spleen, where we're actually getting a lot of the leukemic cells being formed. Again, here we're giving 3 IV injections, and then we're giving 1 treatment with light, and we can see we extend that lifespan about 30%. How does it work? So this is, again, that in vitro proof after the in vivo. So actually we saw that the indocyanin green calcium phosphate nanoparticles can reduce in a cell viability assay about 30%, 20-25% 95% of the growth of these breast cancer cells in culture. And we add in a inhibitor of reactive oxygen species, basically tumors grow very nicely. So oxygen is important, but it's not the only mechanism. And here we're actually looking at energy density, energy yields as a function of the nanoparticle. Here's the encapsulated material, here's the free indocyanin green, and again, as a function of concentration, we see increased energy density, energy flux as a function of encapsulation. So how is it really working? And this is where we get into the photoimmuno nanotherapy. So there are myeloid depressor cells which basically reduce the activity of the immune system and are downregulated are upregulated in certain cancers. So you don't have the immune system attacking your cancer. So here we've used 7 color flow cytometry, and basically we've come up with 2 different subpopulations of myeloid-derived suppressor cells and kind of look here versus here or here versus here. There's less orange here than these upper 2 panels. This is the indocyanin green calcium phosphate nanoparticle. We have less myeloid-derived suppressor cells. At the same time, this is in the nude mouse model where there's still an active T cell component. We also see the same phenomenon in the BALB/C mice, decreased myeloid-derived suppressor cells. And at the same time, we're seeing an upregulation of natural killer cells, and you'll see the next slide, dendritic cells. We've got a lineage switch of going from myeloid-derived suppressor cells to some of these more active dendritic or natural killer cells. Here, look at this orange right here versus here, and you get about 40–50% of natural killer cell increases. And we also see in the BALB/C model, here's the dendritic cells, and here are the natural killer cells with the indocyanin green calcium phosphate nanoparticles. And I'm just going to end on this slide, because this is where we're really going to take this. Everybody's talking about adoptive transfer. Can you actually do what we just saw in vivo? Can we do it in vitro? Can I take breast cancer cells from the patients, put them into a cell culture dish, give them the calcium phosphate nanoparticles, then give them the near-infrared light, take those cells back into the mouse model, and kill the already formed tumor? And you can see we're starting to see some effects. Here's the MDA cells in culture. They get treated with the calcium phosphate nanoparticles and near-infrared light. They go back in the animal, and the formed tumor reduces 40%. And in data, we really don't have time today because I timed this on the airplane, and that's where we get into the lipids. We saw a specific lipid that is causing this. It's called dihydrosphencanine 1-phosphate. And if you want to see the slides, we can actually get adoptive transfer down to about 80–90%. So basically, oxygen is important, but what oxygen does is it changes the way lipids are metabolized. You start forming some of these immunoactive lipids, dihydrosphencanine 1-phosphate, and you turn on the immune system, and you get a lineage switch from myeloid cells to more active lymphoid cells, and you can kill the tumor from the outside. So with that, nanotechnology has the potential to deliver the promise of light-based therapies, not only imaging, but eventually therapy using light. With that, Brian Barth is my senior postdoc who's leading this project. This is his team. James Adair is co-founder of Keystone Nano, which is helping us commercialize the technology. Here are some of the people in his lab, as well as Peter Eklund and Peter Butler, our card-carrying photophysicists, who basically do all the fluorescence correlation spectroscopy. The leukemia data is done by Tom Loughran, who's director of our Penn State Cancer Institute, and Dr. Smith and Dr. Liu, who work for Dr. Loughran. And again, I want to acknowledge I'm co-founder of Keystone Nano, and the conflict is that Penn State technology is being developed by Keystone Nano. With that, I'm open to questions.
Bravo, Dr. Kester. Yes, Dr. Lee?
I wonder, could you comment if the metabolized lipid or if the oxidized lipid is responsible, could the lipid itself be an immunomodulator that you could administer as a drug rather than necessarily?
You've asked a good question. I'm so glad I brought the extra slides. Go. Segue.
Okay, thank you. I'll take the answer sitting down.
I'll go to one slide. By the way, that's the derivative of the lipid. It's a ceramide. That's the Starship ceramide. We had a phosphate. We take away one of the fatty acids. That's your drug. And if we go to right there, so we've taken, here again, a cancer cell, and we've added the magic lipid, and basically, we actually can take these myeloid-derived, in this experiment, we take the myeloid-derived suppressor cells, we actually purify them, we add this magic lipid, and then we actually show by flow cytometry they form into B1 B cells, and basically they'll go back into the animal. There's the growth of the tumor. So with that said, is that the therapy? Maybe. But right now, imaging and using the infrared to get imaging and the ability to do some theranostics at the same time, that's the therapy. But this could be the future therapy. You're absolutely right.
Thank you.
So can you comment on what the limits are on the sorts of other cargoes that you can incorporate? I mean, you've talked only about small molecule fluorophores. Could you do a nucleic acid, oligonucleotide, small protein, et cetera?
So, excellent question, and there are limitations. Basically, if you're a protein, forget it. We're 20 to 30 nanometers in size, and we can go as far as polypeptides. That's the limit in terms of size. In terms of what we can encapsulate, we can do small molecules from hydrophobic drugs like some of our ceramides, which are selective apoptotic agents for cancer cells, to what was being commercialized now for Keystone Nano, which is doxorubicin. And we want to see if we can make Doxil, which is a liposomal version of doxorubicin, better and have better kinetics when you put inside the calcium phosphate nanoparticle. The holy grail, you're absolutely right, is sRNA. And a lot of companies are working on liposomal technologies to deliver sRNAs, but again, you have to do that in a cationic formulation of lipids, which often leads to hepatic toxicology and or lung toxicology. So what we've done is we can put the sRNA into these calcium phosphate nanoparticles. They have no charge. In fact, they're then highly pegylated. So you don't have the problem of cationic charge as a toxicology. With that said, we're now scaling up to get a therapeutic dose of sRNA into the calcium phosphate nanoparticle, and those are going into animals in the next couple of months.
Hi, Chris Schaffmeister, Temple University. Now, correct me if I didn't understand properly, but if you deliver these calcium phosphate nanoparticles they get into endosomes and then they release when the pH drops, you're going to be dumping a lot of calcium into the cell, which is calcium is a second messenger, which...
You got it.
What do you think the effects of that would be?
So in our first two papers, we asked those questions. Because at the end, we're using calcium phosphate. Yes, it's benign. Yes, it's used on grafts. Yes, it's used for bone grafts, et cetera. But still calcium, and it will have an effect as a second messenger. So the first experiments we did was voltage clamping. And we actually saw, then we added the calcium phosphate nanoparticles, that the cell did not change the way it dealt with the calcium. It actually pumped the calcium out as if it was seeing regular calcium, because the actual amount of calcium we're adding systemically is 10 to the negative seventh or lower. So it's a minuscule amount of calcium to the biological environment. And we've also then asked the question, well, what's the worst thing that could happen with calcium? So we said maybe atherosclerosis. So one of the first experiments we published was that we actually took an ApoE knockout mouse, added the calcium phosphate particles, 10 to the 15th particles per cc for 3 months of therapy, and then looked at these animals who are fed a high cholesterol diet and asked what was the fat content at the aortic arch, and it didn't get worse. So those are the type of experiments that have been published to say that this truly is a biocompatible but a biobenign technology also.
Congratulations for the presentation. It was I was impressed last year. Now I'm just jealous. It's great data. Now, talking about numbers, the first experiment in which you were showing the accumulation in the tumor after the initial sequestration by the liver, how much of the injected doses you get there? And also, what is the mechanism for the clearance from the through the bile, and if there is a limitation in size? Because that's a very interesting observation.
So when we did the ADME and we started looking at distribution, for simplifying, 95% of the material ends up in the liver, 5% in the renal system, and that's pretty much it. However, it doesn't lead to liver toxicology. But even though you're lighting up the liver, it's not accumulating within the liver. It's staying in biological fluids. It's got a half-life we've calculated greater than 18 hours. Now, somehow because of the size and the true mechanism, I have no idea. We kind of alluded to this last year, that we really don't know how it's happening, but it's ending up, when we pull out and we actually image the intestine, you can see the intestine lighting up, and we've published that already, and you can, so it has to be going through the hepatic circulatory tree, through the bile, but beyond that, we don't know.
And it's not the autofluorescence of the diet. So
No.
animals are on a special diet.
Often they're not fed. That was the first question I asked, is, so we ran the controls without the calcium phosphate nanoparticles, without the fluoro probe, et cetera, but we actually starved the animals for 24-plus hours, and it's not the food.
Okay, just.
And then we actually pulled out, and we actually,
And you saw.
yeah. Nasty, but we had to.
Okay, Chazi Baranolds again. Just,
And you've shown the same thing.
what?
Didn't you show that? But go on.
I'll show what you will see tomorrow. But what I wonder, based on some of the experience we have with Doxil development, quite some time ago in the late 80s, one of the important questions you have to ask in the beginning, what's the internal volume of your particle?
So, this is the time I wish Jim Adair was standing next to me, because he can actually just...
What's the diameter?
So so we can vary the diameter depending on what we're going to load. These are 20 nanometer particles plus or minus 7 to 10 nanometers. So, and it's a matrix of calcium phosphate, and our material ends
Yeah.
up intercalated within the nanomatrix. We have estimates when we do the fluorescence correlation spectroscopy. Our photophysicists say that we can encapsulate, they're telling us 40 molecules of doxorubicin inside one particle.
Okay.
With
Good.
the
Good luck.
indocyanin
Good
green,
luck.
I know there's a number for it, but
Okay, good luck. You will have to inject so much material that the body cannot accept.
but we're getting 10 to the 15th particles per cc. 10 to the
Okay.
15th particles per cc. So it's a million billion particles. And then actually, when we do our dosing, and we do this by mass spec, we can deliver up to 10 to the negative fourth molar doxorubicin doses. So,
Well, I tell you, based on my experience in human being, not animal anymore, but really human being, we inject 23 times in 14, which is 2.3 times in
Right.
15. We have 10,000 molecule of drug per particles, and that's about the lower limit. So I think it's very, you have to go for, that's what I want to give you advice for drugs that you need much lower amount. Doxorubicin, you need 50 milligram per square meter body, which is
Correct.
about 100 milligram per human being, about my size. Maybe you are a little bit less. So
no,
it's-
I'm getting bigger.
Okay. You know why they make the measurement in square meter? You know why?
Because more,
No, it's because we become,
bigger.
our volume become larger, and that's why they shift to the square meter and not to the weight anymore, to take care of the fat, really.
Right.
But I'll go for something like vincristine, and that you have to give 2 milligram per much lower amount. So then you have a much, much better chance. I think that's one of the first questions you have to ask. What's the load? Can I meet it? And if not, try to find something that will work
Well-
better for you.
Well, what we're seeing is when we compare ourselves, so if a dose in an animal that gives you a therapeutic dose is 10 to the negative fourth molar, we can show effects at 10 to the negative seventh molar because of the protection. So it's not, I know you're shaking your
No,
head.
doxorubicin is fully protected. We get a lot into
Yeah,
the
no, with
tumor, and still,
Yep.
I wish we could do better.
Yep.
Mm-hmm.
And I can tell you, but it's just a kind of thing to think about for you.
Thanks.
Can I have a second bite of the apple here?
Yes, Dr.
Yeah, so my surface area is increasing over time as well, but that's irrelevant to this question. So I'm just curious, so you mentioned earlier that when you neutralize the endosome, the compartment lyses, it pops. Is that a generalized phenomenon?
No, I'm simplifying. I mean, again, it's all we've got some experiments to prove it, but it's really based on a hypothesis. But basically, we believe the phosphate is buffering the endosome, and then the endosome is not going to, the problem is a lot of nanotechnology takes the endosome ride and goes all the way on that train to the lysosome, and then you don't have a biological product. Somehow, we are getting off that train, and we believe it's because the endosome is not maturing into the lysosome, and we believe that when the calcium phosphate, and we've got experiments that have been published with actinomycin D and some other drugs, are basically showing that what we believe is happening is the endosome does not mature into the lysosome. Somehow, it's opening up, breaking apart, and basically out comes your active. And we believe the phosphate's doing it.
Have you considered possibly like these formulations, if that were the case, and you're actually releasing things into the cytosol, could be used as a mechanism for, like in vaccines, to shuttle things between the class 1 and class 2 pathway? And
Exactly.
have you guys considered using your particles as immunogen carriers for
We
that
actually
purpose?
have. The problem is, from a company perspective, you got to focus on one prize type of thing. But we actually have, and we've thought about that idea. We also thought of actually adding an adjuvant on the outside of our calcium phosphate nanoparticles to rev up the immune system, and we're working with there are things we're thinking about in terms of how we can actually make this into an immunotherapy down the road.
Thank you.
There is a vaccine developed by a German guy based on calcium phosphate particle. Jörg, I remember his first name, Jörg Reimann from Ulm. He has actually a few patents on this. You should check for it.
Okay, thank you.
Thank you, Dr.
Very
Kester. Oh, great presentation, and that wraps up our session for today. By way of summary, we started today with a great overview by Dr. Bourge in terms of what would be the practical clinical applications from embedding our bodies with more technology. And I think Dr. Bourge showed us clearly that there were significant savings in terms of morbidity and mortality from embedding our bodies with some of this cool technology. And we had the benefit of the two presentations from the gentleman from the Institute for Molecular Manufacturing that really showed us the ultimate potential of nanotechnology in terms of going beyond the normal cellular processes that our DNA has programmed our body to perform, and taking it much further in the direction of health. And then during the rest of the day, I find that we had a really refreshing mixture of the substrate independence of nanotechnology. You can take nanotechnology from inorganic or organic materials. You can approach nanotechnology from the DNA up, or from inanimate physical elements and physics and engineering, and bring machines into the body, and then many times blend them together, and even further mix it up with some of these references to synthetic biology and its applications that we had during today. So I think one of the most take-home messages from today is that there are many paths to nanomedicine, and that they are not necessarily all purely physical paths, but there are also biological pathways and synthetic biological pathways, and then biomimetic pathways using physics. The other take-home message that I got from today's session is that quite a bit of progress has been made even in the short course of our sessions on nanomedicine and telemedicine, with the great progress that's been made in terms of applications for bone and for ligaments, for the different connective tissues. Great applications have been made in terms of carrier vesicles for anti-cancer and other treatments using nano devices as carriers. And lastly, to wrap it all together, we had, I think, two very good presentations in terms of the precision of targeting of our nano devices and nano agents, either through ex vivo MRI type of targeting, or using locomotive processes from energy generated by the body, be it hemodynamics, movement, kinetics, chemistry, what have you. There seems to be no shortage of ways to power and direct the devices. So in only one day, I really feel we've had a tour de force of nanomedicine, and I thank all of you very sincerely for providing us that. Tomorrow, I promise to be just as exciting. So please feel free to use the extra time to network with each other, or if you have a talk tomorrow, tweak your talk for tomorrow. We'll have a lovely dinner together at 7 o'clock in the restaurant downstairs. So I look forward to seeing you in the restaurant downstairs, and thank you again for a terrific first day.